Hey everyone. Today's guest is Dr. Gary Churchill, who is a professor at the Jackson Laboratory and the co-director of the Jackson Aging Center, which is a Nathan Schock Center of Excellence in the basic biology of aging. Today, Dr. Churchill will present recent findings from his lab showing that fasting and calorie restriction contribute to lifespan extension. So, welcome, Gary, and let's get into the presentation.
Great. Thank you, Michael. Thanks for giving me this opportunity to present my work. I'm always excited to try to explain to people what we do and why we do this. As you said, I'm going to talk today about some results from a recent study that we were really very fortunate to publish in the journal Nature. With the first author is my colleague Andrea Di Francesco. And the paper describes a study that spanned approximately 7 years uh, uh, of keeping mice on the shelf, feeding them different diets and different schedules, as I will explain, and keeping track of how their health and lifespan played out over that period. So this is me. I'm at the Jackson Laboratory. I've been here for 27 years. It's really fantastic. I spent the whole morning cross-country skiing and I am exhausted. So just factor that into my presentation here. What I want to do is I definitely want to focus mostly on the paper, but I want to bracket it. And this is new. I just feel like I've done this talk a few times. I want to throw some new stuff in. I want to bracket it with some of the preliminaries. What did we do before and what motivated us to look at this study and maybe just a little glimpse of where I think it might go next. So um, this is the before. So I'm a geneticist, and I'm really interested in how the variation in our underlying DNA and our genome affects all kinds of traits, things that are related to health, lifespan, metabolic disease, et cetera. And some years ago, I was really concerned that the studies that we were carrying out in mice were focusing on inbred strains. And inbred strains are really powerful tools for doing genetics, but they tend to be, well, they are genetically identical. And many, many large studies often just carry out their studies and report the results from a single strain. And my concern is that results on a single strain aren't necessarily robust. And there are a lot of concerns about reproducibility in science, which is a whole other topic. But, one of the ways that we can help improve reproducibility and generalize results from a model like a mouse to human health is to introduce some genetic diversity. So these two mice they're diversity outbred mice. They're genetically diverse. The two there are probably about second cousins, so they're not unrelated. They're from the same birth cohort. That'll become apparent. And what else can I say about them? It's a really special mice because we took this picture a few days after their fourth birthday, both born about the same time. And a typical mouse lifespan would be 2.5 years. So 4 years, pretty remarkable. And what to say? Um, these were two mice of 900. They were on a caloric restriction protocol, so we had reduced their food intake by about 20%. They eat healthy food. We take care of them. Veterinarians examine them. They lived a good life. In fact, we had them in pens with a large format so they could have lots of friends. They were group housed with about 7 or 8 other mice throughout most of their lifespan. But these two really, made the extra mile. It's not really linear like the dog ears joke that we often hear, but the human equivalent, these mice are easily centenarians. They're well over 100 mouse years old. So let's take a closer look at them. Oh, I should say that we don't normally name our mice, but when you work with these genetically diverse mice, and they're all very unique, they're all different, they're all one of a kind, and you work with them for 4 years, you really get to know them. And I have allowed the technicians to name their mice on their fourth birthday. And on the left here, this is Blanche, the brown mouse, and the black mouse is Grace. So Blanche and Grace. Here we go. Here's Blanche. Well, 4 years old. What can you say? Her fur looks kind of tatty. She's got a hunched back. It's kyphosis. She probably has osteoporosis. There's a nick in her ear. What I find really striking is the hind end musculature is wasting away. So she probably is not getting around very well. She probably can't reach the food hopper. Without a doubt, they're feeding this animal by putting food and water on the bottom of the cage so that she can access it. You also notice there's a kink in her tail. You know, we don't really know what old age is, but we know it when we see it, right? This is an old mouse. This is what she looks like. Her second cousin, Grace, also 4-year-old mouse, the distinguished gray. It's a really nice conformation on her body, smooth tail. The fur looks great, good musculature. This is the 4-year-old mouse you want to be, right? How did she get there? I'm going to say largely luck, because out of 900 mice, Blanche died a few weeks after her fourth birthday, but Grace persisted for 9 more months, and she is still the oldest diversity mouse we've had in the mouse room in now many, many years, and many, literally thousands of mice that we've put on lifespan protocols live to be 4 years, 9 months old. So go Grace. So how did she manage that? Well, we want to understand it. So we've been carrying out some other studies, and this is the published study that Michael referred to a moment ago, where we looked at dietary restriction in diversity outbred mice. The mice that we looked at, there were 960 of them altogether. They were all females. We did that intentionally. I'd like to say, and I will say that there's a really strong male bias in model organism research. A lot of studies are done with only male mice. That's a real problem. It's also not ideal to do a study with only female mice, but if anything, we tipped the scale back a little bit. And honestly, we were concerned with a study of this size and of this duration that male mice could potentially be aggressive, especially if we food deprive them. We've since done these studies, and we're getting better at the husbandry. And I know that our upcoming studies are going to include both males and females. But right up front, please, this is a female-only study. What did we do? Well, we let about 20% of the mice just eat all the food they want. It looks like highly processed food, right? But it's in pellets, but it's actually a grain-based diet. It's really healthy. It's fortified with more than all the things that mice need to stay healthy. So they're eating good, healthy food. And I know people have this image that, oh, the mice are, eating all they want, so they're overeating, and they really don't necessarily overeat. Some do, some get obese. But on average, a mouse is going to eat about 3.4 grams of food per day. And that was our ad libitum baseline. So we have a lot of experience. We know that's about how much they eat. So we put some other mice on caloric restriction. Mild caloric restriction, just like Blanche and Grace, 20% restricted. They get about 80% of the food that they would normally eat every day, and 40% restricted. This is an extreme CR. They're getting 2 grams a day. They'd like to eat 3.5 grams a day. Just picture your plate, okay? You'll see in a moment, the 40% CR is a really extreme intervention. To do CR, you have to weigh the food every day. You have to calculate how many mice are in the cage, et cetera. It's a lot of work. So we thought, well, might be easier just to do intermittent fasting. And we also didn't know at the time whether caloric restriction and intermittent fasting would do the same things. Um, we planned the study way before the 618 style of fasting became popular, and now I wonder we got to go back and look at that. But we decided that 1 day a week with no food or 2 days a week with no food, and we did that by on Wednesday, we changed the cage. The mice are put in a fresh cage, fresh bedding. They can't squirrel away any food. We don't fill the hopper. 24 hours later or 48 hours later, we fill their food hopper and we treat them just like ad lib mice. So they have all the food, hopper's always full. And these are the 5 interventions that we looked at. So it's good. When you talk about dietary restriction, the details really matter. And I'm going to spend 2 slides just to show you, oh, I put these mice on 20% caloric restriction. What does that really mean? Well, this is what they do day to day throughout the week. This is their daily food consumption. This one really puzzled me for a while. The ad libitum mice are eating more food on Wednesdays. It's like, what is happening? Of course, slap your head. This is the day they change the food hopper. So they have fresh food on Wednesdays. They always have food, but it's fresh on Wednesdays and they eat more. So there's that. Here's a 1 day fasting. You can see there's 1 day, 24-hour period. The food is taken away. They have no food. But look what happens the next day. No surprise. They compensate. And by the weekend, they're eating normal amounts of food. And this dotted line, this is the ad libitum average. The 2 day fasting mice, 2 days with no food, and then they also compensate. They continue to compensate through the weekend. And here are the CR mice. You notice their daily input is below where they would like to be, but something happens on Fridays. And this is another just practical logistical decision. I can't have my staff coming in on the weekend every day, every weekend for 7 years. So on Fridays, we said, heck, we'll just triple them up and go home. And the 20% mice would have a feast and spread it out over the weekend. The 40% CR mice did something pretty amazing, not surprising. They ate all their food within a few hours, and then they went 2 days with none. So already it's not so simple. It looks like the 40% mice are also doing intermittent fasting, intentionally or otherwise. And, there's just a lot of fluctuations here, the compensatory feeding. And you can really get down to, these are 4-hour intervals, but you can get down to pretty fine resolution. We put the mice in metabolic cages where we could track everything, how much food, how much carbon dioxide they're breathing, how much oxygen, their energy expenditure, how much wheel running they do. And there's so much to say in these graphs. They're just amazing. A mouse will normally start to feed. You can see here right at the beginning of the dark cycle. Of course, the CR mice start to feed as soon as they get food. You can see the food deprivation period. You can see for the 1 day and the 2 day. You can see the Friday feast. And then by Sunday, all the food is gone. This parameter, RQ, is the respiratory quotient. This tells us what kind of fuel source animals are burning for their energy. If an animal is burning carbohydrates for energy, they're going to have a high RQ value. And if an animal is burning fat for energy, they're going to have a low RQ value. What I find amazing about the 40% CR mice is how they go through this daily rhythm is really striking of completely switching their metabolic base. They're burning fat, they're burning carbs, they switch. It's like they're exercising their metabolism. A little hard to see on this busy graph, but all of the mice have a bit of circadian, but it really gets flattened out with the ad lib mice. Another thing to notice is that RQ plummets when mice are deprived of food, and you can see again the feast and famine strategy. Interesting, the magnitude of the RQ fluctuations at the level of the individual mice don't predict their lifespan. That isn't what I would have guessed. I was really, really hoping, this is the magic bullet right here, metabolic flexibility, lifespan, not the case. Mice who aren't eating have low energy. Make a note. Ironically, the 40% CR mice who are always low on energy are always running on the wheel. They even get up in the middle of the day when they should be asleep and they run on the wheel. And this has been studied in other contexts, and it's a pretty clear indication of food-seeking behavior. They're hungry, they're looking for food. So mice, when they're not getting enough food, they have low energy, they're hungry, and another bit of data that I'm not going to show you, they're cold. So CR, okay. What do these dietary interventions do? Well, no surprise, they reduce body weight. And if you look here on the left, for this first 6 months, all the animals were ad lib. We wanted them to become full adults. Here they are, they're full adults, and we put them on these regimens. I want to add too that we eased the 40% and the 2-day onto their regimens. We didn't just flip a switch here. So we got them onto their restrictive regimens. And here's what an AL mouse would do. It's just going to continue to gain weight until it's about 20 months old. Interestingly, I forgot to mention the AL mice, the 1-day fasting mice who compensate, they don't have any caloric restriction. They eat just as much food as an AL mouse over the course of a week, and yet they lose weight. So same caloric intake, and they're having weight loss. The 20% and the 2-day IF, they're definitely both calorically restricted. They're losing weight. But look what's happened to the 40% mice. Immediately, their weight starts to plummet. It drops down to a certain level, but it just never picks up again. On average, these are population averages. These aren't individual mice. The individual mice are just all over the map. But on average, they're having rapid weight loss. It goes down to what would be a ridiculously low level, below 25 grams for an adult mouse is very low, and they never really pick it up again. So, here they are, they're negative energy balance for their entire lives. I want to just flip this graph because we'll always plot our data in chronological time. But you can plot the data in a different time scale. And it's not perfect, but if we go from birth to death, so we're going to take each mouse's data and stretch it according to their time of death. And we replot the data so that you can see as mice approach the end, you can see they pretty uniformly lose weight. The 40% CR mice, they don't have much to lose, but they are also losing weight at the end. I just want to point out this proportion of life lived scale is not a perfect surrogate for biological age, but I think it works pretty well and it's going to give us some insights, and I'll come back to it. So I wanted to show you in terms of body weight how that looks. Dietary restrictions increase lifespan. That's in the title. You knew it was coming, right? Here are the ad lib mice. Their median lifespan is about 25 months. The 40% CR mice, those mice who are cold and tired and losing weight, they live 9 months longer median, and about 34 months. That is amazing. The IF mice live longer. The 20% CR mice live longer. Now, when I say this, there are a couple of caveats I just want people to keep in mind. I know, Michael, your audience, very health conscious people thinking about diets, thinking about their caloric intake, and probably thinking about what this is going to do to my lifespan. So if you look at the lifespan data, same data, just plotted a little differently. Lifespan in months. On the x-axis, I've got the different dietary intervention groups. And each of these points is telling me at what age a mouse died. And I want you to notice that the 40% mice, they're not all living long. Here's the median. So it's definitely, the odds are better. And here's the tail. So those very extreme mice, Blanche and Grace, right out here. And I want you to notice, by the way, how many 4-year-olds we got out of this study. We got a bunch of 4-year-olds. We're running low on on names, but I just don't want people to think, oh, I'm going to restrict my calories and I'm going to live longer. If you restrict your calories to this extreme, the equivalent of what we've done for the mice, you will not be happy and you probably won't live remarkably longer than a mouse who just ate what they felt like eating. It's still amazing. Okay. I just don't want to give, first of all, it's a mouse study. Everything I say has to do with mice, but we're always anthropomorphizing and thinking, whoa, what does that mean for people? Well, I'm pretty sure the meaning for people is pretty much the same message that you're getting here from the mice. Caloric restriction has some benefits. I'll show you in a minute, it has some detriments. And it does enable a lucky few mice to live a really extended lifespan. So there you go. Does intervention extend lifespan for everybody? One thing, we came in with a hypothesis that mice who were smaller prior to the intervention and mice that were bigger prior to the intervention, first of all, we know they have different lifespans. We know big dogs die young. Same is true of mice. Bigger mice when they're young, mice that are bigger when they're young generally have a shorter lifespan. So would the interventions care if the mice were smaller or bigger? And it turns out that for smaller mice, all of the interventions extended lifespan. The effect doesn't look quite as dramatic here. It's compressed a bit. But for the IF mice, for the heavy mice, the intermittent fasting didn't extend the lifespan on average. So this has also got us thinking that these interventions aren't necessarily right for everyone. And that's a big open question that we're beginning to investigate. Unfortunately, we can't really draw solid conclusions from our diversity mice because every mouse is an individual, and we did not have the opportunity to put two mice who are genetically identical onto the same interventions or different interventions. That's ongoing. Give me 5 years and invite me back. So this body weight thing is puzzling. It turns out that losing body weight has shorter lifespan. And this is what I mean. Here's our body weight curve, and I drew some arrows on top of it to show from 6 months to about 18 or 20 months, ad lib mice are going to gain a certain amount of weight. Each mouse gains more or less weight. Some of them gain a lot. Some of them lose weight, actually. Here's the blue, and here's the 40% CR. They almost all lose weight. So if we look at individual mice and we ask, well, did you lose more weight or gain more weight? And we look across all the intervention groups, the mice that lost weight, the solid line, had shorter lifespans. Lost weight, shorter lifespans, lost weight, lost weight, lost weight. It's not good to lose weight. But wait a minute. The dietary restriction extends lifespan and it causes mice to lose weight. So this is a real dilemma, I think. So this one I've never done. So let's try this. I want to explain a little bit of why body weight. So one of the hypotheses floating around about dietary restriction is, oh, well, you restrict their diets, they lose weight, they don't get obese, it cures their metabolic diseases, and they live longer. That's not true. We put the mice on these diets and they live longer. And you can see that here. This is the same plot I showed you earlier, just turned on its side. Here's a plot of the change in body weight. And just like the other graphs that I showed you, AL mice tend to gain weight, 40% CR mice lose weight. So putting animals on these restrictive diets causes them to lose weight. So that's a negative effect. If we look at lifespan on the y-axis by change in body weight on the x-axis, and we draw the regression lines for each diet, they're all positive slope. So what's happening here is diet causes animals to lose weight, and more weight gain is associated with longer lifespan. So diet can't be explained by change in body weight because the sign is wrong, goes the wrong way. So there has to be, in addition to this, there has to be some other effect of diet that's changing lifespan. And that's what we want to know. And I'll give away the punchline now. That's what we still don't know. But we do know we have some ideas where this might be happening. So dietary restriction is doing something other than weight gain, weight loss, to affect lifespan. In the Dorito study, one of our objectives was to examine health of the animals. And we're really good at measuring mice here at the Jackson Lab. We measured everything under the sun. I just show you the hairball just to give you a sense that, at 3 or 6 time points across their lifespan, we measured hundreds of things. I forget the total number, but there may have been over 700 individual measurements, not including body weight, which we collected weekly, which would be now thousands of measurements on each individual mouse. And we asked, of all these things that we measured, if we calculated their diet-adjusted lifespan correlation, so which traits are associated with longer lifespan, which traits are associated with shorter lifespan. In this little diagram here, here's a trait that I called phenodelta. That's really hands down. It's most strongly associated with very long lifespan. And here's the trait most strongly associated with shorter lifespan. That's the red cell distribution width at 10 months of age. Some of these other traits like myeloid and lymphoid cell populations are immune traits. And I didn't get into the fat content, but when animals are not losing weight, they are maintaining fat, and maintaining fat is definitely a good thing. Positive association with lifespan if you can maintain fat on one of these restrictive diets. So let's pick apart phenodelta for a minute. What this is every year for a month, we sent the mice off to a new mouse room. They get separated from the cage mates. They get measured. We put them in open field. We do blood draws. They go in the NMR machine. It's a pretty stressful period for them. It lasts about a month, and they almost all lose weight. So which mice live longer? So this is their weight change. And this is the same story again, except this is during a very focused period in their lives when they're stressed. So animals, by the way, these are just z-scores because the technical reason it works better on the plot. Mice who lose weight during the stressful period have shorter lifespans. Mice who don't lose weight during the stressful period have longer lifespans. This was really the clue as to what's going on with body weight. And you'll notice it's true across all the diets. It's a little softer here in the 40 percenters, but it's true across all the diets that resilience to weight loss is an indicator of long lifespan. And what I think, this is my opinion, is that these animals across all the diets, independent of which diet they're on, they come with a genetically encoded level of resilience. Their resilience is what's determining their lifespan. Resilience is is a word. It's an English language word. It's a very meaningful word, but in terms of molecular mechanism and what's going on, we'd really like to know what are the molecular signatures of resilience, um, future experiments here. But I think all the body weight stuff can be explained by, oh, I'm stressed. I'm not getting enough food. Can I maintain my weight or can I not maintain my weight? And the mice who are intrinsically capable of maintaining their weight are living longer. Oh, there's some other stuff going on. We measured a lot of things in their blood. And there's this, there are a lot of traits in the blood, both related to your erythrocytes, your red cells, and your immune cells that are associated with lifespan. And I'm just picking on one. It's called red cell distribution width. If you go get a whole blood analysis at the doctor, I'm sure most of you know this, red cell distribution width, RDW, expressed as coefficient of variation, is a number that's going to be on there. And it's very similar to the mouse number. And look at this, mice with high RDW, lifespan on the y-axis, RDW x-axis, mice with high RDW are almost assuredly having shorter lifespans. There are mice who die with low RDW, but mice with high RDW, one exception, right? This could be a mistake, but high RDW is an indicator of mortality. So let's look at RDW predicts mortality. Honestly, I talked to one of the physicians who was associated with the study. And he's like, oh yeah, we know this. Physicians know this. This is why RDW is on the medical report, the blood analysis. So if you look at all the mice together and you just look at their RDW, this is 10 months, negative slope. There's that one. But there are a lot of 40% mice out here, and these are the 40% mice who had short lifespans. They have high RDW. If we go to 20 months, 22 months actually, the reason the plot looks funny is because mice that died before 22 months did not get their second RDW measure. So everybody who was measured is now in the top part of the plot. And you can see again this phenomenon, and then out here at 34. So I picture it, if you had a movie, if you could continuously watch this through time, I just picture these points kind of rolling down here, and then the mice are dying, rolling down and dying. So RDW is a really interesting measure. High RDW, short lifespan. But wait, the fun's not over, because look what how RDW responds to intermittent fasting for 2 days or 40% CR. At 10 months of age, they've been on their intervention for 4 months. This RDW number on average has really shot up. So that's not good, but the slope is less, and we know they live longer. So it does appear that the intervention is doing something adverse to the red cells. And yet the age effect is flattened, and ultimately they live longer. And remember, this is chronological age. I swap this over to the proportion of life lived. And here's something that's really amazing. All these lines are parallel, and they all have this inflection point at about 70% of life lived. RDW starts to go up. Remember the movie. Here we go. These mice have turned the corner. And the fact that the lines are parallel tells me something. These mice are all changing through their biological time scale in a very similar way. And there's something independent that's happening that's just bumping the RDW up in these two extreme groups. That is a weird observation that I'm curious about, but let's go somewhere else for a minute. Okay, I said in the beginning, I'm a geneticist. I'm really fascinated by genetics. I always have been. You can ask my mother about fruit flies in the house when I was a kid. So I really want to understand what are the genetic underpinnings of lifespan. And one way we can do that is we can genotype all the mice and we can do a genome-wide association study. We're going to map loci in the genome that are associated with lifespan. And a mouse has 19 autosomes and an XY chromosomes and a mitochondria. The the height of the peak here is showing me the strength of association of a particular point on a chromosome with lifespan. And the higher the peak, the stronger the association with lifespan. So there's this really high peak here on chromosome 18, I'll come back to. We can also calculate how much of the variability in lifespan is explained by different factors. You remember that lifespan is a really noisy trait. It was even for the 40% CR mice, there were short-lived mice and long-lived mice and a lot of variance. And it turns out that diet explains about 7% of that variance. And then after accounting for diet, body weight, which we talked about a lot, that comes in at around 1%. Chromosome 18, just all by itself, explains 4.5% of the variation in body weight. And the rest of the genetic background, all this stuff out here that really, we can't sift out. There must be dozens of loci that are affecting lifespan. Together, the genetics is explaining about 20%. So if you're a mouse, if you're a DO mouse living in these happy conditions, this is the breakdown. There's a whole other lecture to explain why it's more complicated in humans. But diet has an effect. It's a huge effect, but it's dwarfed by genetics. Interesting that chromosome 18 locus is not only a lifespan, we call them quantitative trait loci. It's not only associated with lifespan, it's associated with RDW. Weird. We have a working hypothesis right now that this locus on chromosome 18 affects something that's closely related to RDW, which affects lifespan. So we have a causal hypothesis and some ongoing studies that I hope will resolve this eventually. So that's the Dredo study. Oh, here's a little synopsis. There were lots of lifespan predictors, diet. Here's your chromosome 18 effect. You can see it's huge. Here's RDW, the lymphocytes, percent fat, which I didn't talk about, but mice with higher body fat are living longer. I think it's a resilience thing. Here's change in body weight that I did talk about quite a lot. So you can just get a sense of the magnitude. The single genetic locus is huge, diet is huge, but look at the shape. All these other factors really don't push the tail, but look at how diet pushes the tail of the lifespan distribution. It's different. Diet's doing something different, and this is what has me puzzled right now. So I just want to look at a bunch of traits. It's called the myeloid skew. How many of your white cells are innate immune versus adaptive immune, myeloid to lymphoid ratio? This is just showing AL and 40% CR. And you can see 40% CR attenuates the rate of increase of the myeloid skew. Sounds good? It is good. But if I change things to PLL, to biological age, these two curves lay right on top of each other. And look, 70%, it's not as sharp as some of the other ones. But on a biological timescale, this ML ratio is tracking biological age really well. Here's our red cell distribution width. Remember, it bumped up in a flattened slope, and we put it on the PLL scale, biological age, parallel lines bumped up. Here's one I didn't talk about, hematocrit. High hematocrit is good. 40% CR is good for hematocrit. It's bumped up. It attenuates less rapidly, but on biological age scale, they're parallel. So what I'm wondering here is whether caloric restriction changes the rate of aging. I'm not even sure what the rate of aging means, but it looks like we're measuring something. And this something that we're measuring has a really sharp inflection point at 75% of life lived, which is really striking. We have some ongoing studies. We're interested in red cell and immune traits. And just because it's on here, I would like to point out inflammation. 40% CR does something amazing to inflammation. It goes down, um, goes up on AL. You can see that just like everything else with these diverse mice, it's really noisy. But if you look on the lifespan scale, sure, it's going down for most of the lifespan. But even the 40% CR mice, when they get to that last leg of life, inflammation shooting up. So things to things to ponder. These are some of the questions I'm really interested in chasing down after this study. And I think that the immune system and the erythroid system are two very likely candidates for revealing mediators so we can better understand how caloric restriction is acting at a physiological or even biochemical level. This was a huge study. I'll just say it was largely funded by Calico Life Sciences. Very grateful, and also the Jackson Nathan Shock Center. There are a whole number of really wonderful collaborators at Calico. I'm just going to point out Andrea, who was with me through thick and thin on the phone every week, sometimes 3 times a week. And also David Botstein, who's been a really wonderful inspiration for me. David is now retired. He's the one who made the study happen. He said, "You got to do this. I'm going to invite you out to Calico. We're going to make it happen." And he did. Some of my folks here at JAX, I'm going to shout out Laura Robinson, who through the entire study was my project manager. Wonderful phenotyping team. And Karen Svensson, who was responsible for Blanche and Grace, collaborators at UPenn and Beth Israel. And oh my God, thank you.
Your study is, from my perspective, iconic. It's going to be looked at for decades. You mentioned translatability. So I have so many questions. I hope we can go through them all. So you mentioned translatability. So the pattern that you saw for positively correlated with lifespan and negatively correlated with lifespan, regardless of which diet that the mice were on. So just using higher lymphocytes at relatively younger ages, you showed in your paper too, so lymphocytes decline during aging. They also decline during aging in people. The
Right.
RDW increases during aging, and higher levels are associated with an increased all-cause mortality risk. So in terms of translatability, sure, it's in, female mice specific to one genotype, but that biomarker pattern is similar in people. And it then raises a question, if you could discover, the recipe, whether it's diet, exercise, supplements, magnitude of CR or not, IF or not, to keep your lymphocytes relatively high and resist that age-related change, and keep your RDW relatively low and resist that age-related change, can you push lifespan and health span even out? So what are your thoughts on that in terms of translatability with the biomarkers?
The measurements that you referred to are really similar between mice and humans. The same patterns of change happen. The numbers are even the same. I know these mouse numbers inside and out. When I go to get my annual physical results, I can read the numbers because they're the same numbers I've read on all these mice. So we're measuring the same things. A lot of the same phenomenon happen. Why do we study mice? Well, we study mice because we want to understand human biology, find ways to improve human health, be translationally, medically relevant. But you're obviously not going to do a study like this with humans, just ain't going to happen. So, and mice give us access to a whole suite of tools that would be inconvenient to use in a human study. So do I believe these results translate? I believe it, yes, but that's my belief. It's an inference. I'm going out on a limb and saying, oh, if this works in mice, it's likely to work in humans. And I think all of model organism science works that way. And ultimately, that's why we end up having clinical trials. And I'm not going to get into the hullabaloo about why clinical trials fail after we've studied mice, except maybe they should have used my mice, but that's a whole other story. Um, can we, well, the rate of aging, as I was showing you at the end, is malleable. We know if there is a such thing as aging, we know that we can change its rate with these caloric restriction interventions. As I said early in my talk, these are really extreme interventions. You don't want to do that, trust me. But it does tell us something really important. It says we can move the needle. Um, well, what do we have to do? And is it even possible to move the aging needle without accumulating all these adverse outcomes, cold body temperature? Um, I doubt the 40% CR mice can reproduce. And I think we raise them in germ-free facility, I think they would be susceptible to infection. So you're going to have trade-offs. Can we find the path, the mechanism, the molecular key point, or the dietary recipe? I don't know. We'll have to keep doing this stuff for a while till
Yeah.
we.
So in terms of the CR mice being more prone to infection, so is so obviously I get in a controlled study, whether it's a controlled feeding study in humans or even on a standard chow diet in mice, that's a variable you want to control. But when considering that, even on CR, or even these genetically long-lived mice without CR, that the last 25% of life, that mortality rates are basically increasing no matter what. What are your thoughts on maybe adding something to the diet? Like, so for example, soluble fiber is, there's a couple of studies showing that it improves survival after influenza infection. So if you infect mice with influenza, you allow them to have, I think it was in the water even, and then, so mice that had soluble fiber in the water, they survive better, right? So what are your thoughts on adding things to the diet that could potentially flatten that, that increased mortality rate for the last quarter of life?
Anything I say is going to be opinion. I will say that the diet that we feed the mice is a grain-based diet. It has a high fiber content, about 5%, which is about double the average American diet. So they're getting a healthy amount of fiber. They have their vitamin supplements in the food, so they have healthy amounts of vitamins. We don't really know what the ideal diet is for old age, and there could be some elements of the diet that could be improved that would increase outcomes in old age. And I think they would be really fascinating to study. And with the amount of bandwidth that any one group like mine has, we're going to have to defer that. I will say that I am very, very interested in this question of do mice on CR have increased susceptibility to infection? Because we see things kind of going both ways. We see at one level their immune system looks good. The lymphocyte proportion's high. They maintain their T cell populations. Their level of inflammation is low, but we don't know. And if the people who are funding the government are generous with us at their next round of funding, we're going to be doing the very experiments that we need to do to ask whether caloric restriction leads to a better late life response to important diseases that affect humans.
Infectious disease or
Yes, specifically Pseudomonas, which is a bacterial infection. You've probably had it and you were fine. But if you were an elderly, hospitalized individual and you encountered antibiotic resistant Pseudomonas, it could be really bad. So we would like to know whether, just like with these mice where we extend their lifespan with extreme intervention, whether when we put them on extreme intervention, we can alter the behavior of their immune system and determine whether we could increase resilience to an infection. And if we can, that tells us that it can be done. It doesn't tell us how to do it in a practical sense for a human, as your question try to address is there something we can add to the diet that will improve our susceptibility to infection late in life? We don't know.
Can I make a bet within that context?
Yeah.
So I'd bet that the ad lib fed mice at very old ages would probably do the best with Pseudomonas. I'd bet that calorie restriction, 40% CR, probably be the first to die off. They just don't have the energy to build an immune, or maybe their immune response is even better. But just to push back on so standard chow, don't get me wrong, is better than a purified diet. It's got soluble fiber, but it's lacking carotenoids, just as an example. So it's alpha and beta carotene, beta cryptoxanthin. I mean, it's got a little bit of beta cryptoxanthin because it's got corn, but lycopene, it's not in there. Lutein and zeaxanthin. I mean, these are consistently shown to be associated with younger biological ages. Um, so we actually did a study where I created a high soluble fiber diet and compared it against standard chow and fed it to aged mice, actually at the JAX lab that we published last year, and threefold higher soluble fiber content. So um, with that in mind, I wonder though, so I'm leaving most of academia. I'm not going to, bang my face against the wall to see if reviewers will fund an animal study up for does a high soluble fiber diet extend lifespan, or higher than standard chow. But
Mhm.
I don't know. Just I have that thought in my mind that what would infectious mortality look like at older ages with a 3x higher soluble fiber content? Um, what do you think?
Fascinating. It sounds like you've already queued up some evidence that it does, and to to get people to eat a 3x increase in soluble fiber diet would be I'm sure people do it.
But the other side of that equation is I've had pushback. So AFRI funded our human study, which is where enrolled. Well, we enrolled our last two participants last week. But the pushback from the NIH, from reviewers, which was even if this works and you get positive, so gut muscle axis, we're looking soluble fiber to impact muscle. Yeah, it's not about me, but just real quick, we got a lot of pushback. Even if this works and soluble fiber, a high soluble fiber diet has a positive impact on muscle, who's going to follow it? Nobody eats fibers as is, but at least it would be a proof of concept that it could work if people decided to do that, right? So,
Mhm.
um, anyway, all right. So I have another hypothesis that relates to your study, and that is and it relates to the body weight. That loss of body weight in all the groups towards the end of life, whether it's the last quarter or last half with the ad lib mice. So when considering that immune function declines during aging, right? Probably regardless of which group, even the 40% CR, they probably have some T cell senescence, for example, albeit not as great as the ad lib, but immune function declines during aging, regardless of group. So body fat produces, adipocytes can produce antimicrobial peptides, cathelicidin as an example. So for me, it makes sense that with a declining immune system, there's an increase in body fat percentage that may be compensatory for the declining immune system, so that the animals who are able to add body fat, which would explain in your study why higher body fat at a relatively younger ages was associated with a longer lifespan. So, what are your thoughts on then it becomes the question of, is this an infection story? So when the system can't add body fat and it loses that resilience, then it's just an autoimmune condition now where it's shedding muscle that may be infected with microbes and shedding fat. So, what are your thoughts on the infection hypothesis as being a part of this story?
I don't know that infection is a big concern in the SPF conditions, but I think your conjecture about the relationship between retaining adiposity and retaining immune function is fascinating. And the data are there. And did I look at that specific dimension of it? No, I definitely think we want to take a look at that. Um, And when mice get to the end of life, they are losing weight. Often, it's very rapid, and it's almost always fat mass that they're losing. And in many cases, with the mice, it's because they have cancer, and their cancer is burning all their energy, and they just burn off all their fat, and that's end of life.
But in the case of many of the mice in your study, I think you mentioned that, or in the paper, it mentioned that there was no obvious pathology, there was no obvious cause of death, which couldn't that suggest an infectious hypothesis?
I'm just going on what we know about mice from having sent them to necropsy, which for a study of this scale was way too expensive. So we did not do cause of death. Um, I think something like 80 or 90% of mice die with a tumor, not die of a tumor, but die with a tumor. Um, can never tell what they died of, but you can tell what they died with. So mice have a high propensity to get cancer, much so higher than humans. And that's a whole other question in itself.
So
Yeah.
just staying on the infectious hypothesis. So there was a recent paper published on IgG, immunoglobulin G, increases during aging, right? It's produced by immune cells. Calorie restriction knocks it by about half. So, and then IgG accumulates selectively in adipose adipocytes, which says to me that it's either an auto CR is protecting against some autoimmune condition, or IgG is produced by immune cells and then it's going into adipocytes because it's chasing microbes and trying to detoxify. So I, I'm still on the infection hypothesis with that, but are there plans to look at markers of microbial burden systemically, whether it's IgG or LPS or even whole genome sequencing in blood or tissues?
Has not been on my radar.
Mm.
Um, we're really uh, keen on understanding how the mice are going to respond to an intentional infection. When I say the mice are in SPF, that means specific pathogen free. So the mouse rooms are clean to the extent that we can identify pathogens that might be harmful to the mice, and we've eliminated them. But no environment is ever entirely free of microbes. Obviously, the mice have microbiome. There are bacteria, viruses, fungi of all types, but of agents that are known to be harmful, we've cleaned them up. So you could very well be looking at the immune system responding to microbes that are not necessarily classified as harmful, but they're going to still elicit an immune response. Um,
I
yeah,
didn't mean environmental microbes. So gut leak increases during aging. So it's
or
possible that what's in the microbiome could leak into the blood, um, and then triggering the immune response. And then if you can't increase fat when the immune response goes down, maybe that's a part of the increased, more dramatically increased, towards the end of life. I don't know, just an idea. So all right, so I have other questions too. So the genetics of longevity. So that's always fascinated me. Are you familiar with Michael Rose's work in Drosophila?
Mhm.
So what are your thoughts on, to me, it makes sense that let's breed, long-lived mice to long-lived mice and do that. And granted, it would take forever, but if there were ways we could evaluate based on biomarkers at 10 months old, like you guys did, or 16 months at a relatively young age, you don't have to age them to 50 months. Could we breed, potentially long-lived to potentially long-lived and do that for many generations and then see really what are the genetics of longevity?
Yes, we could, but I'm going to conjecture that the long-lived mice that we breed would be long-lived in the particular environmental context that they were bred in. And if you look at human causes of mortality, thank God it's going down, but smoking, smoking is a major cause, still a major cause of death by, whether it's lung cancer, heart disease, whatever. Um, if people didn't smoke, it wouldn't be a factor. And there are genetics associated with risk of mortality due to smoking. And those genetic factors, they wouldn't matter. So I think, in a sense, every gene in the genome is a lifespan gene. If a gene had no survival benefit or no survival value, evolution would have let it drop off the wagon at some point. It wouldn't be there. Um, so the genes and the genetic variants that are affecting lifespan, my opinion, are largely context specific. So uh, you know, I'm living in a plastic cage. I'm being fed a high fiber diet. Everything is just so. In that case, this locus on chromosome 18 is a real genetic time bomb for me. If I have the bad allele or the good allele, it's going to have a huge effect on my lifespan. But if you take the same mice and you put them in a different context, I don't know. I can't guarantee it. Um, we've compared several of our studies, and that chromosome 18 thing still hangs around. It makes me think it's pretty transferable, robust genetic effect. But I also believe that there are, hundreds of loci that are active that are contributing to the heritability of lifespan in this context. And that if we just change some details of the context, that those lifespan loci would change as well. And as humans have evolved, if we go back, 10,000 years ago, life was very different. The genes that were beneficial for health and lifespan in that context compared to the genetic variants that are beneficial or detrimental now, some of them have absolutely flipped. Um, uh, yeah, I'm going to have a hard time putting my fingers on it, but some of the effects of genetic variation on metabolic disease in an environment where we have constant availability to high calorie food versus thrifty genes that really helped us when times were lean. You can see that these are all going to be affecting lifespan, and their effect on lifespan is context dependent. So I don't know if that's helpful answer or not, but yeah, we could breed those mice. What would it mean? I'm not sure.
I mean, if it would be evolutionarily conserved. So if you see, Drosophila that have certain gene networks, chromosomes that are linked with longevity, and you also see that in the mice, maybe it suggests that in the people. But then what if you don't have that genetic, those genetic loci? Then it goes to biomarkers. And if it is RDW from chromosome 18, if that is translatable into humans, then it goes back to the idea of what's the recipe for resisting that the age-related change, which would be the best that one could do, right?
Mhm.
Yeah. All right.
I I wish in some ways that I'd taken up I'd stuck with my Drosophila studies from my teenage years. It would be much easier to do the micros experiment than to do it with mice. But I am also concerned that what we learn wouldn't necessarily be relevant. Um, on the other hand, I think looking at these, body weight, body fat, red cells, immune system, these are mammalian traits. Some of them are fly traits. They're all parallel to things we can measure and ascertain in humans. So I think that the relevance of some traits is higher than others. And the role of genetics is, I should say this, interesting, but not central. I think we really want to be looking at the physiology and the physiological health, the immune response, at that level. I think that's where the meaningful translation is going to take place.
And then the things that we could do to keep it as youthful as possible, I guess, independent of what the genetics, your background genetics may include.
Right, sleep right, keep your sexual life healthy.
Right, but it's that striking last quarter of life that just screwed no matter what diet you're on. That's what I'm after now. It's like, if you're tracking biomarkers of all these things, inflammation, RDW, red cells, myeloid, neutrophils, monocytes, lymphocytes, and you can discover that personalized recipe for keeping it low, and you start to see that pattern just deviate at some age, right? Then the question is, what do you do, right? I mean, modern medicine doesn't have an answer for that. at least not yet. How are you going to reduce someone's RDW?
Well, there are some philosophical questions that I'm not going to be able to answer today, and I'm hoping there's still some time before I have to grapple with them myself. We're all going to die, and I think that it the laws of physics, it's inevitable. Entropy is going to catch up, and there are things that we can do that are reasonable. 40% CR is not reasonable. There are things that we can do that are reasonable that will give us a better chance to be healthy as we age. And I think those are the things that we need to pay most attention to. And I appreciate what you were trying to do with this podcast and trying to talk to different scientists and think about the issues. There are a tremendous number of details that we don't understand. Even if we understand in some broad sense, oh, lifespan is malleable, we can restrict calories and we can affect health and lifespan. But when I showed in the beginning, what does it mean to put a mouse on CR? Well, it's a really complicated manipulation. And what is the dietary composition? That's got to be important. Um, we think they're getting a good diet. Maybe they need more fiber. Um, anyway, they're big, open-ended questions, but I think the philosophical question of when you're facing that last 20%, what should you do? That's for somebody else or for me another time. Thanks.
All right, wait, hang on. So hopefully I could push a little bit further into that. Everything you said is great for the majority of the population, right? And I'm with you 100%. Eat well, get good sleep, and eat a normal diet, not too much. I'm with you 100%, right? So what are your thoughts on, and I hear you, you say, you said, everybody's going to die. We're all going to die, right? But don't you think there'll come a point at some point, and I can't say when, I hope it's within my remaining lifetime, your remaining lifetime, all of our remaining lifetimes, that we can say that there is no more death. Maybe that's not the case. Maybe it'll be 500 or 1,000 or 10,000, some number in the future. Will everybody always die? What are your thoughts on that?
I think it is the case that everybody will always die. And it's a necessity because we are so complex. Um, the 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 physics of the amount of energy that it takes to maintain the complexity it is just it's like gravity. You can't deny it. Um, Things will break. We have developed mechanisms to fix things when they break, but ultimately the things that fix the things break, and there's just entropy is going to catch up with us.
For sure.
And it doesn't have to be your opinion, but
Yeah, no worries, no worries. Heat death of the universe, right? Eventually the sun's going to burn out, right? Even the sun will die at some point, but who's to say human lifespan has to be capped at 120 if medical science and research can push us out even further? I'd rather take those extra healthy remaining life years relative to, this 120, which is just a blip. It's just a period at the end of a sentence in billions of years of evolution, right? So
yeah, it's a big question. Is there a wall? Is there a point, a chronological age that we're unable to exceed? Um, I don't know. Um, I would like to think that for as many years as I have, from a personal perspective, we all want those years to be as healthy as possible. And as I said, as you know, as we all know, there are things that we can do that are reasonable to improve the odds that we're going to be healthy for longer.
so
And cross-country skiing for ridiculous numbers of hours probably didn't do me any harm, I hope.
So no CR for you, exercise, regular exercise, no intermittent fasting. Uh, have you integrated
No,
any?
I don't tolerate it well. Um, I eat a good healthy diet. I probably eat too much meat and eat a lot of blueberries. I'm from Maine. What do you want? Um, yeah.
And then tracking biomarkers, are you tracking lymphocytes, body fat percentage, RDW, not paying any attention to it?
No, I get an annual physical. I think that's generally a good idea, in case some warning marker comes up, you want to know. I I know you've been doing this incredibly deep tracking. It's amazing. Uh, I, I I wouldn't have the patience for it.
It's just like CR. Most people maybe don't want to do it, but I love it. I look forward to doing it. It's like my N of one science experiment, So
You got to do what makes you happy,
yeah,
and if you live longer doing it or you don't, doing what makes you happy, well, let's get into the philosophy of it again. I'm trying to live the life that I want to live. And look, I always wanted to be a scientist, and look how lucky I am. I'm lucky and grateful, and so far, pretty healthy.
definitely. All right. So current projects, future projects, one of them being infection resistance. Are there others that are on the horizon that you want to share?
Yeah, no harm. I'm interested in the effects of reproduction on lifespan, specifically female reproduction, whether the whole, um, you know, mating, pregnancy, birth, lactation, these are all really stressful, energy consuming events that will happen to many females, mice or humans. And they must, like CR and IF, impact the metabolic health of individuals and females, women. And I do wonder how that plays out. And again, the gods at NIH need to smile on me, and I will be right on it as soon as it happens. So,
So how pregnancy and going through all those processes that you mentioned, the whole gamut, how that impacts lifespan?
lifespan and particularly late life health, immune function being on my short list. Want to see what happens to that RDW. I have some thoughts about what might happen.
So I'm sure you know, but during pregnancy, neutrophils and monocytes go up like big time, like this big spike, right? So it's a tremendous immune reaction. But I'm guessing would your bet be that the relative to virgin female mice, the the mice that gave birth would live longer or would they live shorter?
Uh, my speculation is that there are trade-offs and that there are going to be genetic predispositions to influence how individuals respond to their reproductive cycles, whether they are in the virgin cohort or the reproductive cohort. And we're going to use we've been working a lot with a new genetic model where we are able to make outbred animals like DOs, but we can replicate the genotype. And so, of course, what we want to do now is look at the same genotype with and without CR, same genotype, with and without infection, with and without reproduction, and ask the question whether on average CR extends lifespan, but is it true for an individual? Because, from the seat you're sitting in, the individual question is the one that matters, right? Should I do this as an individual? It's not a public health question then. I'm asking the public health question with this study.
Nice. Well, looking forward to it and looking forward to inviting you back on the channel to share your work. So thanks again, Gary, and I hope to see you soon.
Yeah, again, thanks for having me. Thanks for giving me a chance to expound on my research. I do love what I do, and happy to share it with you all.
Thanks, Gary. Ciao.
Bye.